studies in support of NDA, ANDA, BLA, and IND applications. I will now turn it back over to our instructors.
Hello, and thank you for joining this session today. My name is Lynda Lanning, and I'm a member of the GLP team in CDER's Office of Study Integrity and Surveillance. Today, Dr. Zhang and I will be discussing some animal rule study cases with you. Just a quick disclaimer, the opinions and information in this presentation are those of the authors and do not necessarily represent the views and/or policies of the U.S. Food and Drug Administration. During this presentation, we'll describe the potential outcomes of an FDA CDER inspection of studies submitted under the animal rule and describe two cases of bioanalytical observations from animal rule study inspections. We will discuss observations from a natural history study and from studies, bioanalysis aspects of animal rule studies. As a bit of a reminder, animal rule studies can be categorized into three general types. Natural history studies are conducted to define the animal model in which efficacy will be evaluated. Animal efficacy studies are intended to provide the primary evidence of effectiveness to support marketing approval, and these studies should be of adequate design and well controlled. And PK and/or PD studies in animals are used to select a dose and dose regimen in humans. Overall, 70% of inspected animal rule studies were conducted in compliance with GLP regulations. When we separate these into study type, 100% of the PK studies, 76% of efficacy studies, 18% of natural history studies, and none of other types of studies were conducted in compliance with GLP regulations. As a reminder, Compliance Program 7348007, used for inspection of non-clinical studies conducting animal rule-specific studies, applies to both GLP and non-GLP animal rule studies. The study type I'll be discussing today is a natural history study in non-human primates. It was performed to support development of a post-exposure prophylaxis treatment, which would later be evaluated in an efficacy study. The challenge agent for purposes of this discussion is aerosolized V. bad bug. And remember that natural history studies are conducted to characterize all aspects of the animal model being studied, everything from the challenge agent specifics to pathogenesis of the disease in the test system. This natural history study was conducted under biocontainment level 3. Experimentally naive cynomolgus macaques of Vietnamese origin were included into a study design where 3 males and 3 females were assigned to groups that would be sacrificed at 4 different time points post-challenge to assess the progression of disease, and a group of 6 males and 6 females that were assigned to a group that would be followed for 28 days and sacrificed then. A target presented dose was 1,000 CFU, and animals were challenged on study day 0. Scheduled euthanasia was on days 2, 4, 5, 6, and 28, as discussed above. Now let's take a moment and review the study objectives. Remember, this is a natural history study, so it was designed to determine the natural course of disease in cynomolgus macaques of Vietnamese origin challenged with V. bad bug. As is often true for natural history studies, this study was designed to try to identify a biomarker as a reliable indicator of disease. And very importantly, this study formed the basis for the animal model as being predictive of the disease pathogenesis in humans. This is, of course, a requirement for animal models under the animal rule. Assessments included pathogen load in both blood and tissue, body weights, clinical observations, telemetry monitoring of temperature, respiration rate, and heart rate, clinical pathology assessments, necropsy, and histopathology. The presented dose of the challenge agent was measured to be 183 to 6,300 CFU for the serial euthanasia groups and ranged from 43 to 627 CFU for the day 28 euthanasia group. Remember that the target presented dose was 1,000 CFU. Clinical observations were given scores based on severity, and scores began to increase on days 2 and 3 and progressed in those animals that proceeded to moribund euthanasia. Scores were used to determine moribundity based on the protocol-defined euthanasia criteria. Following the protocol-defined euthanasia criteria, 6 males were euthanized in the day 28 group in moribund condition on days 6, 7, and 8. 1 female animal survived to day 28, whereas 1 was found dead on day 8, and 4 were euthanized in moribund condition on day 9. The 1 surviving animal received the lowest challenge dose of the study, which was 43 CFU. Telemetry was used in this study to help identify a possible biomarker for disease. Respiration rate increased 48 hours post-exposure, disassociated from the circadian rhythm, and remained elevated in those animals that proceeded to moribund condition. Heart rate increased 49 hours post-exposure, dissociated from circadian rhythm, followed by a decline in those animals that proceeded to moribund condition at about 144 hours post-exposure. At about 47 hours post-exposure, it was noted that animals had an elevated body temperature or fever defined as an increase at or above 2 degrees Celsius above baseline, and a disruption of the diurnal body temperature pattern. And this appeared to mark a shift toward disease in these animals. These changes in body temperatures were consistent among animals and provided evidence showing a potential biomarker to determine when pulmonic disease shifted from an exposure state to a pathological state, and then progressed to a declining state that resulted in morbidity and eventual death in these animals. You might think that heart rate and respiratory rate changes, as described above, could also be considered biomarkers. However, remember that both heart rate and respiratory rate are heavily influenced by environmental stimuli and other factors like dehydration, so they are less attractive as a reliable biomarker. All animals were shown to be bacteremic between days 3 and 8, and the bacterial load in the blood increased in proportion to disease progression. Bacteria were recovered from all tissues examined, with the highest amount found in the lung. The 1 animal that survived to day 28, scheduled necropsy, did not have bacteria in any examined tissues. Remember, this particular animal received that lowest challenge dose of 43 CFU. Hematology findings included an increase in total circulating white blood cells, primarily due to increases in neutrophils and monocytes. And this increase peaked by day 2 post-exposure. There was a marked increase in C-reactive protein between days 2 and 3 post-exposure, and by days 4 to 5 post-exposure, there was an increase in creatinine kinase and lactate dehydrogenase. On histopathology assessment, it revealed a progression in severity of lung lesions from day 2 to day 6 post-exposure. Inflammation and necrosis in the spleen, regional lymph nodes, and liver appeared on day 6 post-exposure and increased in severity on subsequent days. There was a resolution of pulmonary lesions in the animal that survived to day 28, and it was noted that the microscopic findings in these challenged animals were similar to those observed in humans with this disease. So let's take a moment and think about whether the study objectives have been met.
So here we are again at the slide that presents the study objectives. And the first objective of this natural history study was to determine the natural course of disease in cynomolgus macaques of Vietnamese origin challenged with V. bad bug via aerosol. The study did include this test system, this particular challenge agent, and it was delivered by inhalation of an aerosol. And study animals had assessments at adequate intervals to evaluate trends. So it does appear that this study did accomplish this first objective. The second objective was to identify a biomarker as a reliable indicator of disease. And as you recall, I mentioned that the telemetry obtained changes in body temperature were consistent among study animals and provided evidence of its use as a potential biomarker to determine when pulmonic disease shifted from an exposure state to a pathological state. So it does appear now that the second objective here has been achieved. And the third objective, and remember, this is, of course, a requirement for animal models under the animal rule. The third objective was to show that this animal model was predictive of the disease pathogenesis in humans. And assessments of the animals through the disease progression in this study, along with necropsy and histopathology assessments, appear to be adequate to determine that this disease pathogenesis in this particular test system that was similar between this animal model and that observed in humans under natural conditions. So based on the study outcome, it looks like these study objectives were achieved. So let's go on to the next slide and learn a little bit more about this particular case.
So as we discussed, the study did successfully model aerosol exposure to V. bad bug in naive cynomolgus macaques of Vietnamese origin. The pathogenesis of the disease in the test system correlated to human disease in clinical signs, hematology and clinical chemistry changes, and in histopathology findings in the lungs, liver, spleen, and lymph nodes. And fever, defined as greater than or equal to 2 degrees increase in body temperature over baseline measured by telemetry may be used as a real-time trigger for intervention initiation in future efficacy studies. An inspection of this study was performed per Compliance Program 7348007, and this inspection reviewed personnel, quality assurance, facilities, equipment, operations, animal care, testing control articles and challenge agents, the protocol and study conduct, records and reports, and the archives. During this inspection, the following data was not available for review. Raw data documenting the challenge exposure to animals, raw data documenting the enumeration of bacteria in the challenge agent dose and in biological samples from the exposed animals, raw data documenting the clinical observations, and raw data confirming the identity of the challenge agent.
So here we have a study that appears to have met the study objectives, as we talked about a little bit ago. And it looks like it would be a natural history study that would provide support for use of this particular model in future efficacy studies. Excuse me. However, the CDER FDA, during the CDER FDA inspection, which a data integrity audit revealed that raw data documenting various aspects of the challenge agent exposure, bacteria enumeration in the challenge dose and in biological samples from the animals exposed to the challenge agent, and clinical observations were unavailable for review by the inspectors. So consider how this inspectional result affects the ability of this study to support its use and the use of this animal model in the future. Think about how does it draw any of the reported study results into question? And also, does it affect the data quality and or integrity of the study? Remember, that's what the compliance program it has as one of its goals, to examine the study and records and assess whether it is has data quality and or data integrity, any issues in those areas. So let's go to the next slide for the rest of the story.
In this case, a Form FDA 483 was issued with two observations. The first observation was that not all raw data generated as the result of a non-clinical laboratory study were retained. And second, the testing facility management failed to assure that challenge agent had been appropriately tested for identity. And in summary, the data forming the basis for this natural history study's conclusions could not be verified. Here we have the references used for this portion of the presentation: Compliance Program 7348007 and the Animal Rule Guidance for Industry. And now I'll turn the presentation over to Dr. Cynthia Zhang, who will discuss the case studies regarding bioanalysis. Thank you.
Thanks, Dr. Lanning. As you mentioned, there are various types of animal rule-specific studies. After hearing about the natural history case study, let's switch gear to discuss the pharmacokinetic (PK) and the pharmacodynamic (PD) animal rules studies. These studies were conducted in animals to select a dose and a regimen in humans. As indicated in the 2015 guidance, product development under the animal rule, it's required that PK and PD data for the investigational drug be sufficient to permit the selection of a dose and regimen expected to be effective in humans. The assays used for measuring drug concentration in the appropriate body fluids should be validated. The drug exposures associated with efficacy in the adequate and well-controlled animal efficacy studies should be determined. And the PK information from affected animals should be compared to PK information from healthy animals to determine whether the challenge agent-induced disease or condition affects the PK of the investigational drug. And regarding assay validation, the 2018 guidance, Bioanalytical Method Validation, informs the development of bioanalytical methods used for non-clinical studies that require toxicokinetic or biomarker concentration data. It applies to bioanalytical procedures such as chromatographic assays and ligand binding assays that quantitatively determine the levels of drugs, their metabolites, therapeutic proteins, and biomarkers in biological matrices. The common method validation parameters include calibration curve, quality control samples, accuracy, precision, and recovery, selectivity and specificity, stability, and dilution effects, among others. Now let's take a look at the case study of an animal rule PK study conducted in non-human primates. The challenge agent was infectious bacteria, and the therapy or the investigational drug was an antibiotic drug X bioinhaler aerosol. Concentration of drug X was determined using a high-performance liquid chromatography HPLC method in non-human primate serum. During the inspection, it was observed that the analytical run acceptance criteria of quality control QC samples was not specified in the SOP. As a result, analytical runs were accepted regardless of how many QCs met the acceptance criteria, which was within plus/minus 15% of the nominal values, as shown in the table here. Please note that these are mock data intended for illustration purpose only. It was also observed that during assessment of long-term matrix storage stability, LTS, at minus 80 degrees, calibration standards and QCs were prepared at different time points, as shown here in the table. Now, let me pause the presentation and give you a few moments to discuss what potential objections could be. Good morning, everyone. Please feel free to put your thoughts, comments, and answers or any questions in the chat box, and I will go through them before revealing the answers. Thank you. All right, I see someone was asking to move back to the previous slide. Can we please do that? Okay, perfect. Thank you so much. Yeah, I hope this helps. This is the mock data for the assessment of the LTS. All right, I have seen comments coming up in the chat box. Someone was mentioning the fresh calibration curve to assess stability QC test samples and QC criteria was not met. And QC criterion was not defined in the SOP to follow the BMV guidance, and also not up to the guidelines and standards, and is at risk of disqualification. Those are great answers. Thank you guys so much. So let's give the rest of the audience maybe like a few more moments before we move on to the answers. Thank you. Okay, I have also seen some people commenting on the dates of preparation and about the freshly prepared calibration curves. Also, someone was mentioning a baseline for the temperature and the possible temperature excursions. And 2 months gaps between calibration curves and QC preparation. Yeah, those great answers. And I see one question coming asking why are all dates in the future? As I mentioned, indicated in the slides, those were mock data for illustration purpose only. Even though those would be the future events that have not occurred yet, you could actually deduct the time gap by comparing the dates. So very good thoughts. What else? And I have seen mentioning of, yeah, again, freshly prepared QC and also calibration standards. Wonderful, wonderful. Yeah, I'm so glad to see all those great answers. Thank you. Now, let me pause the presentation and give you a few moments to discuss what potential objections could be. The first observation is that the SOP did not define the acceptance criteria of quality control QC samples for analytical runs. As a result, analytical runs were accepted when less than 67% of the QC samples were within the ±15% of the nominal values, and all less than 50% of QCs per level were within ±15% of the nominal values. In general, the analytical run fails if the calibration and all QC acceptance criteria are not met. The second observation is that calibration standards were not freshly prepared for assessment of long-term stability of drug X during method validation. Long-term stability of the sample over a period of time should be equal to or exceeding the time between the date of first sample collection and the date of last sample analysis. The storage temperature studied should be the same as those used to store study samples. Long-term stability QCs should be compared to freshly prepared calibration curves and QCs. Now let's look at another case study of animal rule TK study conducted in rabbits. The challenge agent was infectious virus, and the therapy or the investigational drug was antiviral drug X by oral administration. Concentrations of antiviral drug X was determined using a liquid chromatography with a tandem mass spectrometry LC-MS-MS method in rabbit plasma. During the inspection, it was observed that hemolyzed samples were received during sample analysis as shown in the table here. However, hemolysis effect was not evaluated during method validation. Again, please note that these are mock data intended for illustration purpose only. It was also observed that freeze-thaw stability was established for 3 cycles during method validation. However, a few samples were stored and retrieved more than 3 times during sample analysis. Now, let me pause the presentation and give you a few moments to discuss what potential objections could be. All right, so I guess now it's for this different case, please take a moment to gather your thoughts and feel free to post your questions or answers or comments in the chat box, and I will go through them. I think there was an earlier question regarding the solution stability. Is there any consideration to exposure to light? That's a great question, and that depends on the drug product. So if the drug product is light sensitive, then definitely there should be additional consideration. But if not, then that may not be a concern. And that's something I guess the lab or the CRO should have been put into consideration during the method development and the method validation. Okay, and for the second case, I see that people mentioning the hemolysis. Yes, that's definitely one of the big concerns. And I see that folks are talking about hemolysis is inevitable and need to have a test for it in the method validation. Yeah, those are great answers. So, and folks are talking about the freeze-thaw testing should cover anticipated freeze-thaw cycles. Yes, definitely. If there are more cycles, actually occurred during sample analysis than what has been established during validation, then actual validation of those additional free flow cycles would definitely needed to cover those additional cycles. And someone mentioned the matrix effect. Yeah, definitely. Hemolysis and lipemic effect were definitely something you would like to consider during method validation, especially when you actually observe the hemolyzed samples being handled. Then of course we wanted to know if hemolysis had an effect on the matrix and also the accurate and precise measurement of the concentrations. People also saying data integrity questionable due to hemolysis and free flow cycle issues. Yep. That's pretty much the answer. Right. So basically, I think you guys got it. That's pretty much the two observations that we had. All right, let's resume. The first observation is that hemolysis effect was not evaluated during method validation, while several subject samples were hemolyzed. Selectivity of the method is routinely demonstrated by analyzing blank samples of the appropriate biological matrix from multiple sources. Depending on the intended use of the assay, the impact of hemolyzed samples, lipemic samples, or samples from special populations can be included in the selectivity assessment. And the second observation is that cycles of sample storage and retrieval exceeded the freeze thaw stability established in method validation. Assays of all study samples of analyte in a biological matrix should be completed within the time period for which stability data are available. If sample handling conditions are changed or exceed validated stability data, then the stability of the sample should be established at the new conditions. I hope the case studies help you better understanding the animal rule specific studies. Please see a list of references where you can find additional information. With that, I'd like to thank you for your attention, and Dr. Lanning and I are happy to answer any questions you may have.
So thanks to everyone that's been entering your comments and questions in the chat box. We really appreciate the interaction. And I just wanted to take a moment and remind folks some of the information that was provided yesterday with respect to the Animal Rule Compliance Program. And just remember that that compliance program is designed to evaluate data integrity and data quality in these animal rule studies. And it applies to animal rule studies that are intended to be GLP compliant or not. So just to keep that in mind when you are thinking about animal rule studies and compliance. And I just see one additional question. I'm just looking to make sure I have captured everything. And there was a little confusion, I believe, with respect to the challenge agent versus the test material. And for the example that I provided earlier, the challenge agent would be the V bad bug. So it is the agent that is causing the disease, the disease under study. So that's the challenge agent. And then the test material would be used in the efficacy studies that are being studied in the efficacy study to see if they are able to prevent or treat disease, depending on what the product indication is. So I'll turn the mic back over to Cynthia for her comments.
Yeah, thank you so much, Lynda. I think I got some questions a little bit more general. When results where actual samples are outside of the validated parameters, can the data be used if additional validation is completed after the fact? So in general, the method validation should be mimicking what conditions or handling of the samples going through during sample analysis. But we all know that things happen in reality. So for example, you would have some samples you need to repeat, you need to repeat after or exceeding the established cycles, free flow cycles. Then in that case, it's always recommended to conduct additional validation experiments. I think in general, there are always opportunities to have a supplemental method validation reports, or sometimes they call it addendum number 1, number 2, especially when there's a change of storage duration. For example, the long-term stability was initially established for a year. Then later on, you realize that, oh, we actually need 14 months instead of 12 months. Then those 2 extra months, somehow you have to conduct the additional assessment to cover up that gap. There's also questions asking if hemolysis is not observed for the study samples. Is there any need to establish hemolysis effect? I think that's a great question, and I would refer you to the latest version of bioanalytical method validation guidance document, which is published in May of 2018. Ideally, as mentioned before, the method validation should be mimicking the sample conditions during sample analysis. So by not conducting hemolysis effect during method validation and just hope the samples wouldn't be hemolyzed during the actual collection, I may not personally agree with that decision, because the way of validate bioanalytical method is basically to predict what could have happened during the actual sample analysis and try to cover all the aspects, right? But again, like I mentioned, I know things change, and we all understand that, but as long as the amended validation report or supplemental or addendum report covers all the conditions of the samples going through, that would be still proven precise and accurate. So we can trust, or where the concentration data would be reliable and the data integrity would not be impacted. And also I saw questions regarding the BMV guidance. As of now, we are still basically referring to the BMV guidance for our practice. The M10 is likely to be implemented in future. However, We do not have a specific timeline yet, but we will definitely announce it when M10 becomes effective in future.
And I just have one final thought, and I just want to encourage folks to remember that the quality and integrity of the non-clinical studies submitted under the animal rule are very important, and they have a direct impact on public health as a whole. So just take a moment to think about that and remember that when you're performing these studies. Thank you.
All right. So thank you to both Dr. Lanning and Dr. Zhang for the excellent presentations. Thank you to our audience for the great questions and discussions. Now we'll take a break and resume at 11 o'clock Eastern Time for our next case study. So